All right. And Jason, you want to take it away?
Thanks, Stefan. Just a quick sound check. Everything good? All right, here we go. So I'm just going to set the stage for Scott and Tressa and give you an update on where we're at with the Hazelnut project, where we've been, and give you some sense of the work that's going to be happening or is starting to happen, particularly around germplasm improvement. So the Upper Midwest Hazelnut Development Initiative was launched in 2007. No staff, no money, just hopes and dreams. And luckily, we've been able to keep things moving. And the challenge of any hazelnut development project is simply time. It is a good solid year, 10 years on a breeding cycle. And so trying to do this is a challenge for sure. But so far, so good. And we've made good progress over the years. Our primary focus has been on germplasm improvement, but along the way, we've had an early adopter community that we've been working with that actually predates our work. Hazelnuts have been a thing, if you will, in the Upper Midwest since for sure the early 1990s, and you can trace it back in the Eastern US back to the early 1900s. So part of working with those early adopters is an outreach education program. We've been developing a brand new way to grow hazelnuts based on a hedgerow. Most hazelnuts worldwide are grown as small trees. We're looking at a shrub row system. We've also been working to build harvesting and dehusking capacity because the production system is novel, so too needs to be the equipment to manage it. And then we've also been working on the market development side because as we know, it's easy to develop, well, easy, but to develop new germplasm, send it out there and hope that something happens, but we know that never works. It ends up in a boom and bust. And so we've been trying to be really intentional about aligning markets, processing capacity, and production all along the way. So to that end, we also operate a processing line. I'm not going to talk about any of these main things. These are available if anyone wants to follow up through our website, midwesthazelnuts.org. I'm mainly just going to talk about the plant material itself. This is European hazelnut, Corylus avellana. It grows in zone 6, 7, 8. Well, I should say zone 7, 8, sort of a Mediterranean climate. It is more northern adapted, but in terms of production regions, it's pretty much confined to zone 7 and 8. It's a small tree. It will produce fine in the Pacific Northwest and the Willamette Valley, but if we try to grow it in the Upper Midwest, it typically succumbs to winter injury and a disease called Eastern filbert blight, which is endemic to our region. So despite there being so many great cultivars of European hazelnuts out there, none of them, the existing ones, have worked in our region. There is a breeding project underway out in New Jersey and elsewhere that is developing potentially more northern hardy germplasm, but again, none of it's been proven. So for the most part, we wish we had plants like this, but we don't. All right, so the other side of the hazelnut spectrum is Corylus americana. This is a plant that grows throughout northern Wisconsin, particularly anywhere you see sand. You're going to have hazelnuts as one of the dominant shrub species. It has, for the most part, been ignored as a commercial crop opportunity because the nuts are so small. We cracked out lots and lots and lots of American hazelnuts over the years looking for improved plants. And you can see, though we've got some really interesting kernel out there in terms of shape, color, flavor, oil content, all the other attributes we might want, the nut size has generally been really small. And when you start to crack things out, it becomes pretty inefficient. Tonda di Giffoni is one of the European cultivars, and so you can just see a size comparison. Anyway, so American hazelnut has lots of potential, but it's more or less been underexplored. And our work continues to look at potential cultivars coming straight out of the wild. And then certainly, as we'll talk about here in a second, through a breeding program. Okay, so the interest has been in the Eastern US and the Upper Midwest is to combine the traits of both species. So the winter hardiness and disease resistance from American hazelnut with the European hazelnut's larger nuts and thinner shells and higher productivity. And what we call hybrids, not true hybrids in the sense, but interspecific germplasm. So we didn't do this first. This was done by private breeders, primarily Badger Set Research Corporation and Forest Ag. I should say most recently working with material that came from private breeders before them. And a lot of this material went out to growers, perhaps unsuspecting. They've maybe thought that they were getting better germplasm than they thought. But because they were all seedling origin, the diversity is really incredible, and the average yields of any of these blue dots has not been high enough to support commercial production. Lots and lots of plants out there, but none at the scale or the quality to really move an industry forward. And it's sort of sat dormant for a good decade. Many of these blue dots still exist though, and enough blue dots of plantings like this, where there is a fair amount of nut production, and there's enough interest and enthusiasm among early adopters that they're willing to put in any level of sweat equity to try to build an industry aligned with the germplasm improvement. In other words, they sort of know that this plant material isn't good enough, and yet they can still go out there, grow it, and harvest a lot of nuts and make product and sell it. Will they make money off of it? Probably not, especially if they're doing full cost accounting. But it gives you some idea of what's out there and the enthusiasm among growers to make this industry work. So we've been helping them, right? Even though maybe nobody's making any money, the fact that they want to do this, the fact that by creating market demand, by creating processing capacity, that makes it possible for folks to put improved germplasm and continues the demand for that project. So I'm going to play a short video here that shows the current state of our pilot processing line up in Ashland. If you hear that hum in the background, that's actually our processing line at work. I'm at our facility right now. This is roughly a 2-minute video, but just so you can see it in action. So when we started, there was no nut processing capacity in Wisconsin. So if you were growing your hazelnuts, you had nowhere to go because there is no nut industry. So we built this from scratch, and it's a little bit of a, an extension. We meet the needs of our clientele, but there's also a little bit of build it and they will come ethos in the hazelnut industry. And so now we have a facility that at least can turn in-shell nuts into kernel in a commercially viable way, less than $1 a pound cost, labor cost to do this. So we've set the stage for growth. We've created an opportunity now for growers to grow the material, get it processed, and get it to market. And that market initially anyway is the American Hazelnut Company, which is a grower-owned processing and marketing company owned by those blue dots that I saw or put on the screen. And they've got some really high quality product. And they're at a point where they're going to sell out their 2022 crop by the end of the first quarter 2023. Great. We have a processing facility and we have a market that has more demand than supply. Okay, so what's been the bottleneck? The bottleneck has been germplasm. So all those blue dots, it turns out, is highly diverse, the plant material that's out there. So from a grower standpoint, kind of a problem, but from a plant breeder standpoint, it's a golden opportunity. And the growers themselves did the work of deploying those breeding populations. So all of these square or triangles are the hybrids. The squares are some European selections, and the circles are the American hazelnuts. And so this is a principal component analysis of genetic diversity, and you can see we're working with we have something to work with, in other words. And Scott will talk more about the work that's extending from this particular project here in a second. So we've got a diverse population to select from, all those blue dots. And so we went through, chose the top performing plants, put those in replicated performance trials starting in 2009. And since 2017, we've had our top plants, what we call our first generation selections. These are in propagation. 5 of the 10 have provisional patents, and there are 4 nurseries currently with PMTAs to do the propagation work, and we anticipate first sales in 2023. So it's a sort of this risk question now for growers, right? So we have plantings of this material at this scale, but nobody's growing this plant material, 10, 20 acres. And so who's going to be the first one to get out there and take that risk and try this brand new crop? So what we're trying to do is create what we're calling a Go First Farm network under this Hazelnuts 2.0. So these plantings combine breeding. So the border rows and pollinator rows are offspring from controlled crosses or half sibs from our first gen selections that will be evaluated over time to look for this next round of improved germplasm. And in between those rows are the cultivars that we've developed, the first gen selections. So by working with local partners on these Go First farms, we're able to de-risk some of this initial work. We certainly have growers that are willing to take on the risk themselves, but we're trying to, again, be strategic about how we do this. The other thing that the Go First farms will do is create geographic clusters. It's way easier, and you're way more likely to be successful growing hazelnuts if you have neighbors growing hazelnuts, because you can share harvesting equipment, processing equipment, and input expenses. And so we're trying to facilitate or enable that through these Go First Farm networks. So the other piece of the puzzle is harvesting. You can hand harvest hazelnuts, but not for very many of them. And so we've been exploring and developing improved harvesting equipment. This is an olive harvester that we own. It's too big, it's overkill for what we needed to do, but it does, from a proof of concept, demonstrate that mechanical harvest of hazelnuts directly from the shrub is possible. This is not, that's probably too loud for you. This was not done before we started working on it. And now we're starting to see even growers in Oregon look at mechanical harvest with over-the-top straddle harvesters. So the technology is there. We're working to optimize the equipment, bring the cost down, and again, try to get these out to growers to help with lower those barriers to entry. All right, so my last slide, and I'll turn it over to, I believe it's Tressa, right? I forget if it's Scott or Tressa, but most of our work to date has been basically search and screen of populations that were created by somebody else. For the most part, open pollinated populations. And that's from that work came our first gen selections. The work we're doing now that Scott will talk about, what we refer to as second generation germplasm, is actually much more intentional, where we are using genomics to identify parents, to identify traits that we want, and combine them through cross pollinations and evaluations of full SIB progeny. So we expect to make, rapid gain through the first gen simply by going from seedling plantings to clonal. We'll see a big increase in productivity and quality of the kernel. But eventually, through this first essentially round of trait-based breeding or actual breeding, we expect to see even more rapid genetic gain. So it's a really exciting time for hazelnuts. This is one of these second generation progeny plantings that we've started to make some selections for, but we are over the last couple of years have established even larger populations from controlled crosses that we've been making. So it's an exciting time, and there's a lot going on right now. So I'll stop sharing and hand it over. I don't know if in the transition here, if there were a few questions.
Yeah, thanks, Jason. Really interesting. Yeah, if anybody has a quick question or two, we've got a few minutes to answer those. Otherwise, Scott, I think, are you next?
I'm happy to go next. I don't know that we actually set a final order, but I can be relatively quick here and then turn it over to Tressa to talk about more specifics about one sort of particular phenotype that we're really interested in. So let me share my
Yeah, if you have any
screen.
questions, you can put them in the chat or just unmute yourself.
Okay, are you seeing this, Stefan, in the correct view?
So we're seeing your current slide and the next slide.
Oh, you're seeing the presenter view. Okay,
Yeah.
hold on a second. How about now? Is that
Perfect.
looking better?
Yep.
All right. Great. Thanks. And yeah, thanks, Jason, for really setting the stage there, giving a very yeah, it's always exciting to see Jason's presentation over the years and just see how much progress is happening actually extremely quickly on a number of fronts. So my name is Scott Brainard, and I'm a postdoc in Professor Julie Dawson's program. And the focus of my research is on developing genomic tools to improve our ability to efficiently breed and select for traits and breed new varieties of hazelnut. And here in the title, I emphasize nut traits, which I'll show a little bit about the work that we've been doing with regards to this sort of suite of traits from a morphological perspective. And then Tressa will follow and talk in more detail about some chemical analyses that we have started to perform, specifically looking at oil content, because this is a very oil-rich nut that has a lot of potential in those kinds of markets. So I will skip over a lot of these introductory slides since Jason has set the stage quite well, but just to kind of reiterate the point that we are growing primarily interspecific hybrids in this in the upper Midwest, and the actual parentage of any one of these particular varieties is largely lost to time, but there definitely is both European. There are European parents in the pedigree of these varieties that have been selected. But I should emphasize that American hazelnut itself is an interesting species from a breeding perspective with lots of room for improvement. So when we think about breeding, I think we can think about both improvement of American hazelnut on its own, and whether as a species or a crop that could be developed in its own right, or as parents for crossing with Avellana, or the improvement of the sort of existing hybrid hazelnut material that is being grown. And Jason already really went over this in detail, so I will skip the whole kind of premise behind the kinds of traits that we're attempting to combine in these interspecific hybrids, as well as this is just another photo of some of the existing first-gen material that has come out of the on-farm selections and is now being tested at scale. So like Jason sort of alluded to, selection and breeding historically has been done purely based on phenotype. So private breeders and more recently the universities have identified high-performing plants on the basis of how much they seem to be yielding, whether they appear to have high-quality nuts, flavor. So you actually have to grow these plants to maturity and see the traits that you are interested in, score those traits, and then make selection. And in perennial crops in general, but particularly in hazelnut, a woody perennial, that's a very long timeline and a very resource-intensive proposition. It could be many years before the hazelnuts fruit at all, flower, produce a crop of any kind, and it can be up to a decade before they reach something that you might call physiological maturity, where you could really say, okay, I can measure the actual yield potential of this plant. Genotypic information can be collected much earlier in a breeding cycle. It could be collected as soon as the plants produce leaves, and you can get DNA out of them. And sequencing is becoming extremely cheap. And so the whole premise behind including genomic data in a perennial crop breeding program is we can better inform selection decisions, and probably most importantly, really increase selection intensity before these plants go out in the field and somebody has to invest land and time and money in growing them for a decade. We can do selection on very large progeny families at the seedling stage. And how you actually use genomic data in a breeding program can take multiple forms, and I'll talk about one of those today. But that's sort of the overarching premise and motivation behind this work. So before getting into an example of using genetic data in a breeding program, here's just a little bit of background on one of the traits or set of traits that we are interested in, and it's these morphological characteristics of the kernel and the in-shell nut. And we're interested in this because it is an important trait, as Jason was mentioning. A lot of people have discounted American hazelnut because it has a smaller kernel than European. It doesn't necessarily need to be a bad thing. Consumer tastes can evolve over time, and markets can be adapted to different sorts of crops. But I do think that being able to understand the genetics of an obviously a critical part of the crop and select for what you do ultimately want is important. This is also a trait that's probably under limited environmental control relative to other traits we might be interested in. So it's a appealing target to sort of test out these uses of genomic data. So we've been using this digital imaging approach where we take photos of a subsample of nuts and kernels from a given bush. Nuts are shown here in shell on the left, and then crack them out and put the kernels back in the square, and we take photos, and then we use just some sort of basic image detection algorithms to measure automatically the size and the shape of the nut and the kernel. That allows us to get not just the total size, but also the percent of the nut that is kernel, which is an important trait, not just from a consumer perspective, but it's a very direct component of yield. And here are some histograms of some of the data that we've collected thus far. I'm showing here in two different colors, two different populations, one being bushes that were just grown, they're being grown in Barneveld. This is the salmon shade, and they are bushes from the DNR. So these are wild, not really wild American hazelnut, but unselected for sure, and more likely to be pure American hazelnut than anything else we had access to when I started. In the Minnesota population is actually an F1, or it's a combination of F1 families that are controlled crosses made by the breeder there, Lois Braun, between UMHDI mothers and pollen from Oregon, from 3 different pure Avellana plants from the Oregon State Breeding Program. So you can see, interestingly, and perhaps not surprisingly, that the distribution is larger for these F1 families that have European like a much greater percentage of Avellana in their background, and also larger, heavier nuts. Although the shape, the sphericity, or you might say just the length width ratio of the nut itself is relatively similar to the Barneveld population. You can see similar things for kernel traits. And the last thing and if you combine these kernel and nut traits together, or in-shell traits, you can get measures of percent kernel. As I mentioned, you can also look at the number of nuts per cluster. And I guess here the kind of important thing is that the percent kernel, which is probably the more important trait ultimately than the actual size and shape of the nuts themselves, are much more similar between these two populations. And so that speaks well for, I suppose, the potential of American hazelnut just on its own. The last thing I'll show is a principal components analysis of these traits themselves. So this is a kind of complicated figure to just interpret intuitively, but basically these different arrows are showing, are each represent a trait, and vectors that are pointed perpendicular to each other are negatively correlated with each other, are non-correlated with each other, and vectors that are pointed at 180 degrees from each other are negatively correlated. And this shows a few interesting things. One is a common observation that the more nuts per cluster you have here on the left, the smaller the nuts and kernels are. And that's a very commonly observed phenomenon comparing American hazelnut, which has many nuts per cluster, but they're small, with European hazelnut that might have 1 or 2 nuts per cluster, but they're quite large. The other important thing here is that the gravitometric measure of percent kernel is relatively uncorrelated with kernel and nut size. And this is, again, I think, kind of just reflecting that fact that although American hazelnuts have smaller kernels and nuts, they don't have necessarily a lower percent kernel. Okay, so that's a bit about nut morphology and just the kind of phenotypes that we're looking at. And now I'll show some results from an F1 linkage mapping population. This is a kind of use of genetic data where you take, well, I'll explain it in a second. The first thing I'll mention is that for this study, we actually had to develop a new genome assembly for these 2 plants called Rush and Winkler. These are historically important American hazelnut accessions held by the NCGR in Corvallis, Oregon. And we built chromosome scale reference assemblies for both of them in order to use, in order to align what are called short genetic reads to this genome and then identify polymorphisms. So this is a bit of an aside here for people who are interested in genome assembly. We did successfully assemble 2 new genomes for American hazelnut. And the way that we used those genomes was to study these F1 populations. So an F1 is what we call a full sib family descended from a controlled cross. And in this case, the parents of this cross are the same parents I was mentioning before with the Minnesota populations. They're a maternal parent from the Midwest and a paternal parent from Oregon State. And these are outcrossed heterozygous parents. There are no such thing as inbred lines in a self-incompatible species like hazelnut. And that means that you can observe recombination events in the F1. So and this leads to a segregating population in the F1, but also the ability to essentially correlate the variation in phenotypic, the phenotypic variation that you see because of this segregation for traits in the F1 with known or identifiable recombination events between the 2 parents. And this allows you to ideally trace back and associate phenotypic variation for, say, nut size with a specific position in the genome, which we would call a quantitative trait locus, or a QTL. So this is one way of trying to build genetic tools for this sort of next generation of selection, as Jason has mentioned. So I'll just show the results from one of these F1 populations, where the paternal parent is this historically extremely important C. avellana variety called Jefferson, and the maternal parent from the UM-HDI is a plant called Eric 4-21. And this is one of the varieties that is being trialed at scale now by the UM-HDI. And there were 93 progeny planted in Minnesota. I think this is actually should be 2015. This population was replicated, the cross was replicated over a couple of years. And for those of you who are interested, the marker development was performed using GBS libraries, where we digested the DNA with enzymes and then sequenced these fragments on an Illumina at quite high coverage. We used about 10 million reads per sample. And then we called indels. So we identified insertion deletion polymorphisms from these short reads themselves with a program called STAX. And again, we aligned these short fragments of the DNA to a reference assembly and then looked for insertion deletions. And we did that as opposed to looking for single nucleotide polymorphisms, because in an F1, you have 4 haplotypes segregating, 2 from one, you have the 2 haplotypes from the paternal parent, the 2 haplotypes from the maternal parent. And if you use a marker type that it can have up to 4 alleles, then you can actually observe recombination in both parents. So that's a bit of a technical aside, but it does turn out to be the case. And this is the kind of genetic map you can construct in an F1 when you use these multi-allelic markers. Very little inflation, so the total map distance is more or less in line with what you would expect biologically. So this is an ordering of genetic markers along the 11 chromosomes. Here I'm using chromosomes. In linkage mapping, you call these linkage groups, but we're using the terminology from the reference assembly we built. And we know the distance between these markers. And we can then do what's called linkage analysis, where we associate phenotypic variation with alleles or genotypes at each of these marker positions. This is comparing the genetic map to the genome assembly, showing relatively good concordance between physical and genetic distances. I won't really get into that. What I want to do is just mention the actual linkage mapping that we did was using this package called FullSibQTL, which implements a type of linkage analysis called composite interval mapping. It's maybe a somewhat complicated linear model if you've never seen it before, but essentially what's being modeled here are the additive effects of each parent. So this is essentially the value of the paternal parent, Jefferson, and the maternal parent, or Eric 42-1, as well as the specific, what they call intralocus interaction, which is the dominance that you would observe at a given locus. So this is the model that's being fit at many positions across the genome, and you're looking for significant additive and dominance effects on the phenotype Y, which is phenotypes are going to be these morphological nut characteristics that I showed pictures of earlier. And I'll just show a few of the results, which are quite striking, I think, that there are actually very significant, not only this is a LOD profile where this is essentially, you can think of it as a p-value on the y-axis, logarithm of the odds, and points above this dashed line are considered to be significant associations between this position in the genome, here expressed in centimorgans, and the trait, which I'm going to show a few of these just for a few representative traits. Here is the height, or the one of the dimensions of that in-shell nut. And these are the estimates of the parameters, so without getting into too much of the sort of nitty-gritty around the numbers, this total R² of the model of 50% using only 4 QTL is actually quite high for these kinds of studies, I would say, especially for traits that we would expect to be quite polygenic. And we see this also for, here I'm showing kernel length width ratio, so the sort of circularity, or we could infer the sphericity of the kernel. Slightly lower R², but again, not negligible. And then this very important trait in my mind, the total percent kernel measured gravitometrically. Again, we see about 40% of the variation being explained by these 3 positions in the genome, these 3 peaks in the LOD profile. So that's really a pretty, that's really kind of maybe brushing over a lot of nuances behind linkage mapping, and might be a bit confusing if you've never seen these kinds of studies before. But just a few conclusions that we can draw from this kind of study is that, first of all, certain morphological nut characteristics, despite being quantitative traits, may be under the control of major effect QTL, which is very promising for marker-assisted selection. As I'll explain, I'll have another comment about that in a moment. The utility of actually selecting for markers as opposed to phenotypes will definitely depend upon specific crosses that you are making. Whether this is a particular QTL that's showing up in a cross exclusively between an Oregon State variety and a Midwest variety, or if it will be robust and stable also in like pure American or pure Midwest hybrid germplasm, we don't know that yet. And I should also say that there's lots of other ways of using this kind of this exact kind of GBS data in association analysis and genomic prediction models. And those have quite a bit of potential as well, and we will be evaluating them. I'll just end with these two photos. On the left, well, I'll say on the right here is one of these variety trials at scale, sort of go first farms of first generation material that Jason mentioned. This is being grown on land owned by the Savannah Institute out in Spring Green. And on the left are new F1 families. And I think this is sort of like the past and the hopeful future of hazelnut breeding. The past here is material that was selected entirely based on phenotype and has shown, I think, tremendous promise. I think there's varieties here on the right that have commercial viability in the Midwest, but it took decades to get there. And if we want to shorten that timeline going forward and make even faster progress with future selections, it would be great to be able to say, take the progeny from some of these F1 families on the left, be able to genotype them at the seedling stage and select for individuals that we know have a high chance of having a specific kernel size or a specific yield, potentially even. Or as Tressa will talk about in a moment, another trait that we can throw into these kinds of predictive models is oil content, and that's another really important trait for a sort of oil seed crop like hazelnut. So that's sort of a glimmer into what we're up to in the development of genetic tools. And I'll just thank all the funding for this work. I'm a postdoc funded by the USDA NIFA program. The Arnold Arboretum funded the development of the reference assemblies. The Grantham Foundation is funding these plantings in Spring Green. And obviously, a lot of partners in this work, including not only the Upper Midwest Hazelnut Development Initiative, but University of Minnesota, who made these crosses, and then a ton of people in the Dawson lab who have been doing a ton of work on the phenotyping side, taking photos and grinding up kernels. And Tressa is one of those people. So I will turn it over to Tressa now.
Thanks, Scott. Can you all hear me okay?
Yep.
Yep, we can hear you.
Awesome. Sweet. Let's see. There we are. Okay, so I'm Tressa. I'm an undergrad studying agronomy, and I work in Julie Dawson's lab. And last semester, I had the opportunity to complete a project on the effects of storage time on hazelnut chemical properties. And through this project, I'll also be able to talk about some of the nutritional contents of hazelnuts. So the data I'm presenting was collected by Marissa and I. Marissa is a technical expert in the Dawson lab, and she was a great help during this whole process. We were able to collect and analyze percent oil data, NMR data for fatty acid content, and we completed combustion analysis for percent protein. Our methods were provided by Peyton Higgins. She's a chemistry graduate student in the Buehler lab. We tested the stability of whole nuts in a cooler for 3 months. So the first round was cracked out and processed in the second week of February, and the next round was cracked out and processed in the second week of May. For our oil extraction process, this took place over the span of a week. So we would crack out our samples, and then we would grind the nuts, and we would have to use liquid nitrogen so that the oil didn't cause them to clump up too much in the NutriBullet. We would then add liquid hexanes and tilt to mix to dissolve. Then we would centrifuge to separate the liquid and solid and remove the supernatant to a new tube. And this was repeated a total of 3 times. At the end, we were left with 1 vial of oils and 1 vial of de-oiled hazelnut grounds. And that's how we could get our percent oil data. So as you can see on the left graph, according to our averages, we did see a statistically significant difference between the February oil percentages and those of May, but I decided to look further into this. Obviously, we don't want to see that difference. We want our samples to remain stable. So if you look on the right, Marissa's values did stay consistent. She had a p-value of 0.94, but mine saw a definite decrease. And I looked back on the experimental notes to maybe see how this could be. And it said that Marissa had to step away from the lab for about 30 minutes. So her first extraction sat in solution for that much longer than mine. And meanwhile, in May, at that point, we had completed this experiment several times. So I definitely became more efficient. And I think that might have been a timing issue where perhaps in the future we should add time onto the clock in completing the step where it's dissolved in the hexanes. So we're hoping to continue this next semester as well, and we'll pay more attention to that. So hopefully we can have a more solid answer on how stable that is. Next, we used NMR spectroscopy. NMR uses molecular physics and crystalline structures to identify organic molecules, such as the fatty acids that we were looking at here. This involved using our oil extracts in the vials. We would add chloroform D and stir it to dissolve. We would transfer this to an NMR tube, and then in the chemistry building, we would run the NMR. And then we would retrieve the data and run it through Mastronova software. That's the image that you see on the bottom here. And we integrate the peaks and finally produce percent distribution data in Excel. We did have some assumptions during this step, one of which is that linoleic, oleic, and saturated fatty acids are the only three categories of fatty acids present in the solution. And then another assumption is that the fatty acids will not degrade in the extracted oils when they're stored in a freezer. But both of those assumptions, Peyton helped us to ensure that we could be confident that those are true. So here are some general information about the fatty acid profile of hazelnuts. Linoleic and oleic acids are both considered to be heart healthy. They decrease the bad cholesterol in the blood. Linoleic is considered to be an essential nutrient. Our bodies cannot produce linoleic acid, so we have to consume it, and it is important for our health. Oleic is produced in our body, and it is also the most widely occurring fatty acid in plants and animals. But it's still considered heart healthy. And then finally, in small quantities, we did see some saturated fatty acids, which are considered unhealthy, and palmitic and stearic fatty acids are the types that you can find in hazelnuts. So the graph to the right shows our average distributions of the three types in orange for February and then blue for the second round in May. Keep this graph in mind when I go to the next slide, because I'll be showing the distribution for all of our samples. We used 8 samples, and Marissa and I each ran those both times, once in February and once in May. So there'll be lots of data shown here. So on the left, you can see the data from the samples that I processed, and on the right are Marissa's. Both sets showed no statistically significant change in this percent distribution. Again, orange is the February samples and blue is May. So therefore, we can be fairly confident that none of the 3 fatty acids degrade at a greater rate than any of the others, and hopefully they do not degrade at all. And now we can see this presented in milligrams fatty acid per grams of kernel. This is where the percent oil content comes into play again. And again, you can see on the left with my values, there was a great decrease, and Marissa's on the right stayed consistent. So again, my hope is that this difference is due to my samples having less time to dissolve and not an actual issue with storage. I decided to include a graph of the fatty acid profiles of some other nuts that we processed since we have the data available. It's interesting to see like how different the distribution of fatty acids are in different nuts. The 3 on the left are 3 different types of acorns, and then the 4 on the right are hazelnuts that we acquired from different sources. The hazelnut profiles do align really well with the samples that we processed. And the acorns, you can see, have more unhealthy fats in them. So finally, we used combustion analysis to determine percent protein. We just had to prepare the samples for analysis, and then we sent them to the Jackson lab to finish that process. So we have our data from the February samples, and our May samples have been submitted to the Jackson lab, but they haven't gotten this data back to us yet. They have a lot of student workers, so our assumption is that they just haven't been around to complete it, but hopefully soon we'll have that to compare. But for now, we can see that the average percent protein in the kernels is 17%, and the shell average is 2%. And the reason we went ahead to look at shell protein is because one of the potential uses for hazelnuts could be animal feed. And in some cases, then you would potentially just grind the whole kernel and shell all together. Yeah. So moving forward, we'll be focusing on the Minnesota hazelnuts we received this year's harvest, and we think that a comparison of fatty acids and protein across a longer period of time in storage might be a valuable metric. Hopefully we have enough nuts to duplicate these experiments. There was a drought this year, so we'll see about that, fingers crossed. We definitely want to continue to make sure our experiments are uniform. I think storage should be an easy thing to keep consistent, keeping the uncracked nuts in a cooler, and then the processed nuts, oils, and kernels in the freezer. And since we found some potential confounding variables in the oil content with the differences in timing, we might increase that timing part of the experiment, and we'll just have to keep an eye on that for future extractions. I have a lot of people to thank in the Dawson lab and other labs as well. So thank you, and also thank you all for being here to listen. Yeah, thanks. So now I think we'd like to open it up for questions from anyone and for anyone.
Yeah, thanks, Tressa. That was really interesting. Yeah, so if anybody has questions, please just go ahead, unmute yourself or put it in the chat. We've got a couple questions in the chat already, so we'll just start with those. Jason, this one's for you. The cost of equipment for a processing facility.
The equipment we currently have, it's an acquisition cost for us was roughly $150,000. That does not include the cost of the actual building. We're in a shared use kitchen facility on the campus of Northland College, so that brings our cost way down. In Wisconsin, if you crack out one nut, you do have to do it under a food processing plant license, which requires all of the upgraded facilities. Hazelnuts on a smaller scale are falling within the cottage food laws in surrounding states. And so they have options, some smaller growers to crack out in their garage or whatever and sell limited volumes. But in Wisconsin, we don't have that option. So that's why we need a fancy processing facility. And it looks like the second question too is related to processing. The model that I suspect the Midwest will follow is the one in Oregon, where you have geographically clustered growers that have aggregation nodes. So they bring all the hazelnuts together, and there's often some form of pre-processing. In our case, it would likely be dehusking. In Oregon, because they sweep the nuts off the ground, there's a whole complicated washing step, which we don't have to do because they're coming directly off the shrub. So there'll be some on-farm aggregation around nodes, and then just because of the cost of the equipment and the food safety regulations, we suspect there will be geographically scattered processing facilities like the one in Ashland to service multiple grower nodes, if you will. So we'll see. It's up to the growers to figure all that out, but as of now, our pilot processing facility in Ashland is more than adequate to harvest or to manage the current production. It's not ideally located, but given all of the other advantages of being up here, it's working fine. So we don't really see the processing as a bottleneck at this point. It's more getting the germplasm out to growers.
All right, other questions?
Yeah, I got a question. I'm not a geneticist or a breeder, and it's been a long time since I had genetics in college, but I do remember the northern corn leaf blight fiasco of the 70s and now tar spot in corn. My understanding from Jason is that there's some genetic or resistance to diseases in the American hazelnut. How do you go about preserving that that trait while selecting for yield and the other factors you're talking about? Because our other industries in ag are pretty much bred for yield and ignored, not ignored totally, but in many cases it was the disease resistance, and we paid the price lots of times for that. Thanks.
Yeah, it's a great question, Scott. Maybe I'll start with an answer and then you can jump in. It is a major concern. We have identified resistance loci, but we don't actually have any of the genes identified in conferring resistance to EFB, and therefore we really don't know what the mechanism of resistance is. We suspect in the American hazelnuts that we've got quantitative resistance, but again, we don't know the mechanism. And so making sure that that resistance stays within our selections is a huge concern. We test it. We inoculate the plant material and make sure we test it for resistance. But that's a huge focus of study. I mean, it literally is trying to protect the entire organ industry, is all about protecting from Eastern filbert blight. And so there's lots of work going into the genomics of Eastern filbert blight to make sure exactly what you said, Carl. This is a disease that is not a joke. It wiped out the organ industry once and it could do it again. And so we can't put plant material out in the Midwest that's going to be susceptible. It's got to be able to sustain resistance and tolerance to what's a highly diverse pathogen, which is endemic to the region. So it's one reason why we're really interested in American hazelnut and not just trying to move a couple of dominant resistance traits from American hazelnut populations into European and just have it be overwhelmed with changes in the pathogen. So trying to maintain that quantitative resistance is going to be super important. So Scott, anything to add on that?
Yeah, no, I definitely echo the risk of Eastern filbert blight and the challenge of breeding for its resistance. It's poorly understood. It's, I mean, its pathology is poorly understood. It is a complicated fungus with a multi-year life cycle that can't be cultured on, artificial growth medium. So these programs like in Oregon or in Rutgers, where in New Jersey, where they're trying to grow where they're trying to grow European hazelnuts, they have tons of EFB. It's maybe like the #1 breeding target for them is making sure that they're breeding with very susceptible material in their background. So they have to be extremely cognizant of it. We're in a good position in the Midwest of relying on material that is largely resistant already, and so these sort of seedling screens in the greenhouse seem to be effective in preserving that resistance. But yeah, it is definitely something to be aware of. And then, I guess if the question was entirely about EFB, I'd leave it there. There are other diseases of hazelnut that aren't potentially as damaging, like in terms of killing the entire plant, but other pest and disease issues that we'll surely confront are challenging in general with tree crops, where you can't just like roll out a new variety in a couple of years. And so I think that again speaks to the importance not only of developing genomic tools that can make that breeding pipeline more efficient, but also preserving diversity in, if not necessarily on farm, if we're going to move towards clonal varieties, like at least across the landscape and in repositories and breeding populations and seedling pollinizer rows. These are really important for being able to respond to new diseases, which an industry, if it took off, would certainly face.
Other questions? Scott, I was wondering, like, at what point will your research be, will we be using that to actually like make selections on the seedlings?
Yeah, I get asked that question a lot by people in Minnesota who are making crosses and want better ways of doing it. I would say that we have these sort of preliminary results that I presented today on QTL mapping in specific populations. I'll be repeating that in 2 other F1 populations over the course of the spring here, and also in the Barneveld population that I mentioned, that's more of a kind of American hazelnut background performing genome-wide association. And in all of these populations, you can build just prediction models. And those are the 3 approaches that we are testing. The first 2 being like sort of ways of trying to find specific regions of the genome that you would select for if you wanted a particular trait. The prediction models being just a way of kind of purely predictively, without an understanding of the genetic control, informing selection decisions. So there will be I will have results this year and hopefully this spring in order to sort of weigh which of those approaches seems most practically applicable in the short term. And then, yeah, I mean, that could immediately be put into use, I would say. Lois Braun and Mark Hammond at University of Minnesota are making crosses every year, and they generate more progeny than they can plant. The sort of practical element that I think still needs to be worked out is a cheap genotyping platform for actually, because in order to use this information, you have to, and the advantage is you don't have to grow the plant to maturity and collect phenotypic data off of it. But if you want to make a selection decision on a given seedling, you do need to genotype it. And right now, that's costing us about $50. You can't do that on that many bushes. So we are exploring and need to test and validate cheaper options that would get us, I think, ideally below $10 a plant. And then you might be able to screen, potentially 1,000 or more plants every year and select the best subset of those to then plant out. I mean, $10 per plant is, there's all sorts of simulation analyses you can run to sort of figure out what's the cost going to be over the course of a few decades to like grow this plant in the field and evaluate it, and where does my genotyping cost have to be to justify that, cost, that sort of upfront cost. But I think we can, that's the one remaining like sort of practical bottleneck to overcome, where once we have the genotypic data and the model, we can put those together, and there's no reason to not start using that data as soon, immediately, essentially. But the other thing is that I would say is like a lot of my work is being built on populations that happened to exist when I started my postdoc, like experimental populations that we're treating as, or that we're using for purposes that was never intended 10, 15 years ago when people originally planted these plants. So the other thing that's happening simultaneously is these, the plantings that Jason and I both mentioned that are going in now, new F1s that are being planted with attention to the pedigree and the planting design in a sort of forward-looking way to be able to use those more effectively, be able to control for spatial variation using statistical methods, including right sample sizes of different progeny families so that we'll be able to do this even better the next time. So that when those families are producing progeny, we can hopefully, it's an iterative process where our accuracy will hopefully continue to get better. So I'd say ASAP all the way through to a decade from now. And we will we'll just keep working on it.
Yeah. Cool. Any other questions? All right. Well, thanks to the presenters today, and thanks for everybody who joined. Yeah, 2 weeks from today, we'll be talking about Kernza. So hope to see you then. All right.
Thanks, Stefan.
Thanks a ton, Stefan.
Bye.
Buddy.
Bye.